Good afternoon, students. Welcome to Raj Malhotra IAS Academy. Today we are in the session, current affairs session, 2:30 session. We are going to discuss about topics of science and tech. Two important current affair topic of science and tech. Very important topics. One is your antimicrobial resistance. It's very important for prelims as well as mains. Similarly, second one is about your satellite launch. Current, you can say, satellite launched in 2021 and 2020, 21 and 22. We will discuss 4 of these launches. We will discuss about some technical things, okay, about these launches, and some kind of, you can say, we will discuss about some orbits which are very important for your prelims. Fine. Chaliye. First topic is your antimicrobial resistance. Antimicrobial resistance, AMR. Antimicrobial resistance. First thing is, what is antimicrobial resistance? What is antimicrobial resistance? Antimicrobial resistance. Okay, antibiotic resistance you have seen, okay, how it spreads, science behind these things, how antibiotic resistance activity spreads, how it spread from one bacteria or fungi, or you can say other organisms, microorganisms, to different kind of, you can say, microorganisms. Okay, we will discuss in detail. We will discuss science behind these things. Science, you know, these things about how these spreads, like over the counter, if we take drug doses, incompletion of drug course, or you can say use of these drugs, antibiotics in your animal husbandry. We all know all these things, but science behind these things, we will discuss that. You will not find these discussion in on any platform, because we ignore the science behind these things, behind antimicrobial resistance. Okay, we will discuss these things in detail. Okay, how antibiotic resistance spread, science. We are not talking about the how it spreads in humans, or you can say, it's second thing is, first thing is the myth behind antibiotic resistance. Many student thinks that antibiotic resistance is about human body. It's not about human body, it's about the bacteria, it's about the fungi, which have some kind of resistance against antibiotics or antimicrobial drugs, which kills that organism, which kills that fungi, which kills that bacteria. It's not the resistance of our body, it's the resistance of bacteria, fungi, protozoa against antibiotics or antimicrobial drugs, which we use, which humans use to kill those organisms. So first thing is, you should have a clear cut idea what is antibiotic resistance and how it is different from other kind of antibiotic resistances in bacteria. Antimicrobial drugs is against any microbial, fungi, bacteria, or you can say any microorganisms, protozoa like your plasmodium, which spread your malaria. Getting my point? So first of all, how these things spreads, or how antimicrobial drug resistance spreads, how antibiotic drug resistance spreads. When humans use over the counter drugs, for example, without prescription, we go to the medical shop and ask them about the drugs, or you can say antibiotics, and we take 1 dose or 2 dose and we do not complete that course. Okay, due to that, what happens, there is presence of antibiotics in environment, or you can say it leads to the increase in population of resistance bacteria. How we will see that. Second thing is, in animal husbandry, especially in animal husbandry, poultry, and you can say in cattle feeds, what the owners do, they use antibiotics in their feed, antibiotics or antimicrobial drugs in their feed. What do you mean by this? In their feed they use antibiotics, they use because to increase the production of meat, because the in poultry chickens will be healthy, cattle will be healthy due to presence of antibiotic, but in return what they are doing, they are contaminating the environment with your antibiotics. They are contaminating the environment with antibiotics. Getting my point? And this is one of the biggest source of your spread of antibiotics in environment, and which leads to the antibiotic resistance bacteria, fungi, or you can say antimicrobial drug resistance fungi, protozoa. Getting it? Another reason is your hospitals and research centers which use the antibiotics, and the environment nearby hospitals is kind of having large amount of in their water, you can say sewage, or you can say in their solid wastes, they discard large amount of antibiotic without treatment, which leads to the increase in population of antibiotic resistant bacteria. I'm saying again, the increase in population of antibiotic resistance bacteria or antimicrobial drug resistance fungi. So how these microbes develop resistance against antibiotic? For that, you should know about science behind antibiotics or antimicrobial activities. That's the important part of our lecture. Science behind antibiotic function and science behind antimicrobial drugs. So, science behind these things. The first thing is your what these antibiotics do. They target specific, you can say specific targets in a bacteria or fungi. For example, your penicillin drug, what it does, it targets the cell wall of bacteria. It disintegrates the cell wall of a bacteria, due to which bacteria get killed. In same way, they can target different important enzymes which are very important for the life of a bacteria, fungi. For example, RNA synthesizing bacteria, DNA enzyme, sorry, RNA synthesizing enzymes or DNA replication enzymes, for example, RNA polymerase, DNA polymerase. These drugs target DNA polymerase, RNA polymerase, or any important enzyme of bacteria. What they do, they target these enzymes, and due to this, what happens, the important function which is very important for a living organism, for example DNA replication or protein synthesis, get stuck, or it you can say, what you will say, कि वो उसको बनने ही नहीं देगा, किसको, प्रोटीन को, आरएनए को, डीएनए को. Getting my point? So there are many kind of these targets. We will see. For example, drugs like penicillin, these are called beta lactams. What they do, they target the cell wall of the bacteria. For example, rifampin, it targets RNA polymerase. What is the function of RNA polymerase? It transcripts DNA into RNA. So what will rifampin do? It will stop the RNA synthesis. RNA synthesis stops, means your protein synthesis will stop, means your bacteria will be killed. Getting my point? In same way, tetracycline stops the protein synthesis, and protein synthesis is important for any living organism, bacteria, fungi, for us also. But when we use these antibiotic, for example tetracycline, it will target the ribosome of bacteria only, not our ribosome. So the protein synthesis of bacteria will be affected, not ours. Getting my point? And this will lead to the, this will lead to the death of the organism, bacteria, fungi, anything. This is the way antimicrobial drugs or antibiotic functions. Okay, by blocking important, you can say, reactions or important parts of the bacterial, living bacteria or fungi. So how these bacteria overcome these antibiotics? They can overcome antibiotic attack by different means. For example, some bacteria, what they do, they inactivate the beta lactams. There, for example, in penicillin, there is a ring. In penicillin, there is a ring. It is a ring structure. And what these enzymes will do, they will open that ring structure. And opening of that ring structure will lead to the dysfunctions of your beta lactam rings of any, you can say, your antibiotic like penicillin. That antibiotic will not function on any you can say, the chain structure of that penicillin will not be affected or effective on that bacteria. Getting my point? They will change the structure of your antibiotic. Second thing is, for example, some antibiotic is targeting RNA polymerase. What will the bacteria will do? They will change the structure of this. If they change the structure of this, it will not be recognized by antibiotic. So it will help the bacteria to save itself from antibiotic. In same way, for example, it will change the structure of their ribozyme. Some minute changes which helps them to structure of ribosome, which will help them to survive the antibiotic attack. Getting my point? So these are few examples. So from where these enzymes comes? From where this RNA polymerase, new kind of RNA polymerase comes? These things comes from kind of in their genetic material. They can be, you can say, they can be result of any mutation. They can be due to result of transfer of gene from one bacteria to another bacteria. So we have to see that. So how bacteria and fungi fight back against antibiotics? We have to see that. When I told you germs develop new cell processes that avoid using the antibiotic target. For example, they will deactivate the penicillin. They will open the ring of penicillin, which will not function on your cell wall of bacteria. In same way, they change or destroy the antibiotic. Germ restricts the access. For example, they will change the entry point of that antibiotic. That antibiotic will not enter the bacteria, fungi body cells. Why? Because they have changed the target on which your antibiotic was attaching. Getting my point? Then you have other thing is, they can by using pumps, they can throw out antibiotic out of their cells. They can throw out the antibiotics out of their cells. And another is, drug cannot, if they change the target, drugs will not function on that same target as it used to do before changing the structure. And how these are transferred from, like, in different bacteria, there are different kind of ways, through plasmids, through transposons, and through some viruses. Every information is embedded in some kind of genes, some kind of genes. Plasmid is a small circular DNA present in some bacteria, not all, in some bacteria which have antibiotic resistance genes. Antibiotic resistance gene. And if a bacteria have to survive, what it can do? It can gain a plasmid from any mean, and we will say how a bacteria can get a plasmid. This plasmid have some genes which functions against the antibiotics, which are antimicrobial drugs, which will lead to the resistance of bacteria against that antibiotic. How these bacteria get this plasmid, we will see that. In same way, transposons, our human body, bacteria, other organisms have some jumping genes. Jumping genes means these are transposable or transferable genes, sequence of DNA which can be transferred from one chromosome to another chromosome, from one part of chromosome to another part of chromosome. During the evolution, many of these, you can say, genes are, you can say, especially in human body, are from some kind of bacteria, some kind of viruses especially. In same way, bacteria can also have some transposons. These transposons, some of these transposons have antimicrobial drug resistance activity or antibiotic resistance activities. How this helps? These transposons, when transferred from one chromosome to another chromosome, sometimes they get activated, and due to that activation they produce some kind of effect which makes that bacteria or fungi resistance against different kind of antibodies, antibiotics, different kind of drugs. And in same way, phages, or you can say viruses. When viruses infect some bacteria, sometimes what they do, they insert their own DNA or some kind of antibiotic resistance genes, resistance gene in bacteria, which help the bacteria to survive against the antibiotic attack, which helps the bacteria to survive the antibiotic attack. This is the way. Transduction, when the antibiotic resistance genes is transferred by a virus, it's called transduction. Okay, any kind of gene transferred is called transduction, and through this method a bacteria can get antimicrobial drug resistance gene or antibiotic drug resistance gene. Getting my point? In same way, two bacterias during conjugation, conjugation is kind of your sexual, you can say, reproduction in similar bacterias having same strains. They exchange their genetic material, and in that exchange one bacteria can transfer antibiotic resistance gene to another bacteria. Getting my point? And third thing is transformation. Transformation is horizontal gene transfer. Horizontal gene transfer means, for example, one bacteria was antibiotic resistant, one bacteria, and it was killed, but its DNA is in environment, and the part of DNA which is resistance to antibiotic is transferred to the bacteria, another bacteria. This is horizontal transfer of gene without any living thing. Bacteria was dead, but its DNA was transferred naturally without any conjugation or transduction in the environment from environment to the living bacteria. This is known as transformation. These are some methods through which your bacteria gets antibiotic resistance gene, which makes that bacteria antibiotic resistance. Got this or not? And in same way, sometimes what happens, mutation occurs. Mutation is sudden. What happens? Bacteria grow very fast, and due to fast mutation, within a day millions of bacteria can be produced. And mutation is change in DNA sequence, and that change can lead to the antibiotic resistance. So this is also one of the method. Mutation is also one of the method through which bacteria gets its antibiotic resistance. Got this? Now, fine. Why I am talking about this? This is a very big problem. Antibiotic resistance is very big problem in whole world, you can say. Okay, Indian government has started a red line campaign against the over the counter use of antibiotics. You will find a red line on some of drugs, especially on antibiotics. This red line indicates that you cannot take that drug or antibiotic without the prescription of any doctor, physician. उसके prescription के बिना आप नहीं ले सकते हो. Getting my point? Red line campaign to make the people aware about antibiotic resistance. Okay, fine. Our next topic is about recent launches of ISRO, recent launches of Indian Space Research Organization. So we will discuss few of launches, 4 launches of 2022, 1 in 2022, 3 in 2021, which are important for your prelims 2022. So what are these? First one, which was launched in February 2022, was PSLV C52 EOS4. EOS is Earth Observatory Satellite 4. We will discuss what is this and why this is launched. Okay. So this mission was launched in 2022, in Feb 2022. Okay. So what was this was launched by which vehicle? You should know about the vehicle configuration. This was launched by PSLV C52. What was the, it is a 4-stage engine. PSLV have 4 stages of engine, and this variant was PSLV XL variant. This is one of the powerful variant of PSLV which can launch higher payloads in your orbits, satellite orbits. Okay. So this mission was about launching US4 satellite. US4 satellite. What is US4 satellite? It is a radar imaging satellite. What do you mean by? These are Earth observatory satellite which have some kind of payload which will see out the, you can say, which will release some kind of radiation and collect the data and transmit the data to the Earth center, for example ISRO center in Bangalore. This satellite is for the weather condition, quality images of weather conditions for agriculture, forestry, plantations. This will function in C-band. C-band is between 4 gigahertz, the wave radiations of 4 to 8 gigahertz. This is the radiations or electromagnetic radiations of 4 gigahertz to 8 gigahertz. Okay. With this was the main satellite, which is the complementary satellite for ResourceSat, Cartosat. These all are Earth observatory satellite, and these are launched in, important point here is, these are launched in low Earth orbits, less than 1000 kilometer of height. Fine. Then you have INSAT 2D and Inspire Sat 1. These are experimental satellites launched by, or you can say these were designed by students, experimental satellites. Then second launch was Amazonia 1. This was launched in August 2021. Same vehicle was used, PSLV vehicle was used, PSLV DL variant was used. DL variant was used. PSLV DL variant have 2 step-ons of 12 tons. Step-ons are used for providing more thrust. Step-ons are used to providing more thrust. Okay. So what was the main satellite in this? The main satellite of this launch was Amazonia 1, to study about the deforestation of Amazon forest. This was designed by Brazil and launched for the Brazil. This was the first satellite launched by NSIL, the commercial branch of ISRO. NSIL. This was the first satellite launched by NSIL. Fine. This is a commercial branch of ISRO which launches the satellites for other commercial purposes. The main satellite here was Amazonia 1. Then you have PSLV C50 launch. This launched a communication satellite in geosynchronous orbit. Geosynchronous orbits are around 35,784 kilometers above the surface of the Earth. They take same time 23 hours 56 minutes 4 seconds that Earth takes to rotate around its axis. So you will find, observer will find satellite at the same place on the sky every day. Getting my point? So why this was launched? To launch a CMS-0 satellite into CMS-01 satellite, communication satellite into the orbit. This satellite have payload in C extended band. This have payload in extended C band. Extended C band is between 8 to 12 gigahertz frequency radiations. Like we have visible range is between 400 nanometer to 700 nanometer. In same way, different kind of wavelength are microwaves are which have larger wavelength as compared to visible, ultraviolet rays or infrared rays. These are used for communication. Mainly these are between frequency to 1 to 40 gigahertz. For example, in 5G, your 5G, the frequency is used around 3 gigahertz. The frequency of radiation, the waves which are used have frequency of around 3 gigahertz. For example, for 6G, this frequency will increase up to 70 to 90 gigahertz. So more power, more energy will be there in 6G technology. Okay, so these are basic things about this satellite, and this will cover whole of Indian mainland and Andaman Nicobar and Lakshadweep Islands. So communication satellites are usually, remember this, they are launched in high Earth orbit around 36,000 kilometers above the surface of the Earth. All communication satellites, and they are launched in a geosynchronous orbit always. Geosynchronous orbit, mainly they are launched in a geosynchronous orbit. Fine. This was launched in March 2021. PSLV, this was the first one, PSLV C49 Earth Observatory Satellite 1. This was also launched to study about the Earth observatory satellites have main functions to study about the surface, topography, weather conditions. These are launched in usually in low Earth orbits. It was launched in early 2021. With this, many, for example, this was the main satellite. Okay. US1 was the main satellite, but with this 18 more satellites were launched. 4 were from USA, 4 were from Luxembourg and 1 from Lithuania, and some were from different universities or some experimental satellites of India. PSLV have capability to launch multi satellites in different orbits, multi payloads in different orbits. PSLV, very few countries have this facility. Okay. Another important thing about last year's space research is small satellite launch vehicle. What is the issue? When we use PSLV as a launching vehicle for small satellites of 100 kg, 200 kg, the thing is the cost is very high. The cost is very high. For example, if you have to launch a satellite of 400 kg and the capability of PSLV is to launch a satellite up to 1725 kg, and in GTO or geosynchronous orbit about 1400 kg of load it can launch in a geostationary or geosynchronous orbit. When we have to use it for 100 kg, 200 kg or 400 kg satellite, then it is the waste of resources. It is the waste of resources because it has capability to launch up to 1500 kg or 1700 kg. To save the money, to save the energy, what ISRO has done, ISRO has developed a new launch vehicle, which is very, very important, small satellite launch vehicle, small satellite. Okay, this is a 3-stage, listen carefully, 3-stage solid propellant, means all the, listen carefully, all the, you can say, fuels will be solid. Okay, and this, the thrust produced by this rocket launcher will be less, but it is capable of launching the satellites up to 500 kg in low Earth orbits. For example, satellites for your Earth observation, satellite for your weather communication. These are satellites which are launched in low Earth orbit. Low Earth orbit is the orbit of satellite up to 1000 kilometer above the surface of the Earth. So this small rocket have efficiency. This can be launched on demand because it is easy to assemble, lower cost per kg, because for example, if you are using PSLV which can launch 1700 kg and you are using it for 300 kg, so the cost is more or not. To reduce the time and increase the production rate, and multiple satellite, nano satellites of 1 kg, 2 kg, 3 kg can be launched by this small satellite launch vehicle SSLV. This is in pipeline. This is in pipeline, and especially the commercial branch of the ISRO is marketing it as a ready to use vehicle for any launch. Getting my point? You have PSLV, GSLV for launching big satellites, but the cost of those launching vehicle is very, very high. Fine. Now we have to reduce the cost for smaller satellites, and for that in future ISRO will use small satellite launching vehicle SSLV, which is, I think you will have question in mains or prelims on SSLV because it's a cost saving mechanism. It's very, very cheap as compared to PSLV or GSLV. Fine. Thank you. यहीं सेशन को एंड करते हैं हम. ये 2 टॉपिक थे जिनके वी हैव टू टॉक अबाउट द साइंस बिहाइंड दीज, स्पेशली एंटीमाइक्रोबियल ड्रग रेजिस्टेंस एंड antibiotic drug resistance. Fine. Thank you so much.